Good morning, all of you. Today we are going to see our first practical. Okay. So see here in this first slide, there are certain test tubes are present, petri plates are present. So there are certain things, equipments which is required in microbiology lab. Before going towards our practical, I will explain you uh, what are the safety rules you have to follow in our lab. Okay. So first rule is that you wear lab coat as well as gloves, cap, okay, in microbiology lab along with the lab shoes, or you can use lab slippers also in microbiology lab. Don't eat anything within the lab, or don't drink water also in lab, because as we know the organisms are omnipresent, so they are present everywhere, and certain contaminations, we are working with organisms, so E. coli is there, staphylococcus aureus is there, again there is presence of fungi is there. If the fungal culture is there, it's spread widely. So these contaminations are entered through the food or water into your uh, stomach or within your body, so it is hazardous to us. So don't eat anything or don't drink anything in microbiology lab. When you are working with any culture, please be conscious with the culture. Don't open it in the air. Only open the cultures within the aseptic area. These are the certain lab rules which we you have to follow in microbiology lab, and follow the aseptic area. That is, you just clean the working table and then wipe it with the ethanol and put on the burners also. Okay, so this is the aseptic area which you have to prepare for micro practicals. Okay, along with the test tubes, petri plates, conical flask, then pipettes. Apart from this, there are certain equipments which are very essential for microbiology. Okay, what are they? We will see now. Okay, we will see the first one is autoclave. Okay, we know autoclave very well. See here, this is the autoclave. It is present in our lab, isn't it? You might be seen because your biochemistry practicals are there in this lab. So this is the autoclave. So what is the construction of autoclave? There are the certain lead is present, and on that lead the screws are there. You have to tighten that screw when you keep your equipments which you want to be sterilized within the basket or jacket. So here is the lead present fitted with the clamps, and asbestos jacket is present. Stationary autoclave may be double door or both ends, one for loading and one for unloading. The pressure gauge is there. Okay, the thermocouple for measurement of temperature usually located within the discharge channel in dummy article. See here, it is outlet is also given, and air vent to remove the air before sterilization, it is given. As I told you in theory also, there is a pressure gauge present to check the pressure, and after that there is a presence of the air vent also. Okay, and at the bottom of that autoclave there is a presence of heating coil, and due to that the steam is going to be sterilized. Okay, so steam to be there in the autoclave. Okay, so now we will see what is the principle of autoclave. Actually it is a moist heat sterilization, as we know that. So moist heat destroys the microorganism by the irreversible coagulation process and denaturation of the enzymes and structural proteins. Okay, what is the actual principle of this autoclave? Moist heat destroys the microorganisms by irreversible coagulation process and denaturation of enzymes and structural proteins. Okay, in support of this fact, it has been found that the presence of moisture significantly affect the coagulation temperature of the proteins and the temperature at which the microorganisms are destroyed. Simply, we are applying certain pressure along with heat there. Okay, so we are applying uh, heat as well as steam is there. So Due to this, the equipments which are present within that jacket or in autoclave, it going to be sterilized. Okay, and there are certain time temperature relationship is there. Means suppose we are sterilizing one media, so we have to wait for 121 degrees Celsius, that is 15 lbs pressure is there, and we have to wait for 20 minutes. See here, it is chart given pressure, temperature and time. So this is the ideal time temperature relationship for sterilization process. Okay, so what are the applications of this autoclave? Okay, and how you are going to be working with autoclave? First we will see articles to be sterilized as placed in the basket or cylinders. As the articles means the equipments, they are placed within the jacket, but before placing it into the jacket we have to wrap it with the newspaper and cotton plug. Okay, it is wrapped with neatly. Okay, then door is closed and clamped, heating is started and steam is generated. Air vent is closed after complete removal of air, that is after 4 to 5 minutes, as like a pressure cooker. Okay, and when the pressure reached at the required atmospheric temperature, the sterilization period is timed. At the end of the period, heat is disconnected and autoclave should not be open before the pressure is fallen down. So this is the procedure you have to follow, and when you come in our practical physically. Okay, so at that time I will show you everything, but today we are going to see these practicals on the PPT also. Okay, so what are the application for sterilize the media, culture media, for identification and propagation of pure strain and of the microorganism, to sterilize the various surgical stainless steel instruments that are required for moist heat sterilization. Okay, to sterilize the various types of surgical dressing, gauze, etc., and to sterilize the host of fourth applicators, equipment, solutions, syringes, etc. Okay, so autoclave is used for sterilization of all the pharmaceutical use, all medical use, for microbiological use. For that we require this autoclave. Okay, so this is about autoclave, and these are certain types of autoclaves. See here, pressure cooker type of autoclave is there, common laboratory autoclave is there, then vertical autoclave which is present in our lab, and horizontal autoclave is also there, and large automatic hospital autoclave is also there. Okay, so these are about the uses and application and working of the autoclave. Now we will see what is the incubator. Okay, see here, it is incubator, it is present in our lab. Okay, actually it is regulated by the thermostat. The thermostat is an appliance fitted to the to regulate the temperature inside the oven or a refrigerator, or inside particularly the incubator. In incubator is appliance shaped like a box which maintains the desired temperature inside. As I told you, it is a chamber which is made up of asbestos. Incubator, it is a box container with insulated walls, and a door is fitted with latch to close the door firmly. Okay, and a hole in the center of its roof is there for insertion of the thermometer to read the temperature inside the chamber. The thermometer is provided at the top. Okay, at the top of the autoclave, and then there is and at the base there is a heating unit is there, heated through electricity. On the front of the base there is switches are there to maintain the temperature also. Okay, on the back side is fitted a thermostat regulator is present. Okay, thermostat regulator is present there, and in the center of the front side beside knob is there, and certain racks are provided inside that. We are going to keep our cultures which we want to be inoculated. Okay, so this is the incubator. And what are the use of this incubator, we will see now. Okay, see here, this is the given, it is a principle, it is worked by thermostat. Thermostat is an appliance fitted to be regulate the temperature inside the oven. Okay, as I told you now, just now, it is the structure of this autoclave, sorry, incubator. Okay, and this is the what are the uses to incubate the organisms, to provide the certain time, certain temperature for their growth it is required. So this is the about the incubator, and it is given here also. The incubator have a wide range of applications including cell culture, pharmaceutical studies. Okay, as well as for providing the appropriate temperature to the growth of organism. Okay, so this is all about the incubator. Now we will see the hot air oven. Okay, so what is the hot air oven? See here, this is the hot air oven. It is also present in our lab, and it is used for dry heat sterilization, isn't it? Autoclave is for moist heat sterilization, incubator for providing the appropriate temperature for their growth of the organisms, and hot air oven is for, it is for dry heat sterilization. Yes. Hot air oven is electric device used in sterilization. They were originally developed by the Pasteur. The oven uses dry heat to sterilize the articles. Okay. So what is the principle of this oven? The sterilizing by the dry heat is accomplished by the conduction. The heat is absorbed by the outside surface of the item, then passes towards the center of the atom. Layer by layer, the entire item will eventually reach the temperature required for sterilization to take place. Dry heat does most of the damage by oxidizing molecules. The essential cell constitutes are destroyed and the organism dies. The temperature is maintained for almost an hour to kill the most difficult of the resistant spore. What is the meaning of this principle? We are rising the temperature. It is above 150 degrees Celsius temperature. So at that time, obviously there is a denaturation of protein molecules is there. Okay, so and how the airflow is there, because in hot air oven there is a provision inside the oven, there is a provision of fan. So due to this fan, the heat is transferred within that chamber. So due to high heat, the certain organisms are going to be killed. Okay, and hence the sterilization is occur, as as we saw in our theory also. Okay, so it is consist of the metal rectangular box with the double door having insulated material, that is, it is made up of asbestos coating. In it, small fan is fitted at the top for uniform circulation of hot air oven and heater at bottom. Okay, so how it will be work? Arrange the material to be sterilized loosely and evenly on the racks of the oven, allowing free circulation of the air and thereby even heating of the load. Okay, air is poor conductor of heat, so do not pack the load tightly. Switch on the power supply and control the temperature of the oven adjusting. Okay, thermostat. So we in hot air oven, as we know that the wetted particles or the material which is heat sensitive, we cannot sterilize that material within the hot air oven. Like plastic can should be melt. Okay, or media cannot be sterilized within this hot air oven. So we are going to sterilize the dry equipments or dry glasswares inside this hot air oven, and which tolerate the high temperature. Okay, we are going to keep it in the oven, just increase the temperature, wait for the time, it provide the constant temperature, it is maintained almost for hour to kill the most difficult resistant spore. Okay, so this is, see here, this is again a figure of the autoclave. Okay, sorry, figure of the oven is there. And what are the applications of this or uses of this hot air oven? It is used for sterilized materials like glasswares, metal equipment and powders. It allows the destruction of the microorganisms as well as the bacterial spores. Okay, so these are the three instruments which we require in microbiology lab. There are again more instruments I will tell you in next session. Okay, thank you.